In this discussion, we're going to be looking at the process of meiosis. And as we go through, similar to what you had watched before with the process of mitosis, I'm going to be modeling this using some manipulatives on my desk. Take note as we go through, you'll find that there are notes and questions to accompany this presentation that you should kind of fill in as we kind of work through it. What we see right here, it is a cell. We can see the yellow boundary. This is inside the cell, outside the cell. And I have a simple cell right now. I only have 4 chromosomes or sets of DNA, when in reality our cells would have 46 of these. Now in the process of meiosis, we take one cell with one full set of DNA and we reduce it into haploid cells. So cells that have half of the number of DNA. So we're going to create 4 haploid cells as we go through this process. Now, what we have right here would be considered to be a diploid cell, and that's because the chromosomes have homologous pairs. We can see that we have these two that are the same length, and they have genes in the same locations. And we can see that these two are the same length, and they have genes in the same locations. This is indicative of homologous pairs of chromosomes. Again, homologous chromosomes means that for every chromosome you get from dad, you get a matching chromosome with the same genes on it from mom. So we would have homologous pairs. We've seen this before while looking at karyotypes. Now again, in our karyotypes, we would have 46 total chromosomes, 22 pairs of autosomes, 1 pair of sex chromosomes, and this would be inside every single cell of our bodies. And again, for our simple purposes, we're only using 2 pairs of chromosomes. So maybe chromosome pair number 1 for the big set I had, and maybe chromosome pair number 10, just for the purposes of illustration. Now again, keep in mind that our cells don't always have chromosomes that look like this, right? What happens to these chromosomes during interphase? Well, they get copied, right? And when they get copied, our karyotype looks like this. We still have 46 chromosomes, but now we have 2 copies of this chromosome and 2 copies of this one because it was duplicated. I'm going to illustrate that using additional pipe cleaners. So this chromosome, it goes through interphase. It makes a copy of itself. There's a green bead and an orange bead in the exact same locations on both. And when we twist them, now my chromosomes take on that X-shaped appearance. And I can repeat that for my blue one here, my blue one here, and my yellow one here. So now, I've taken my DNA and I have duplicated it. At this point, before we get further, it may be good to make a few clarifications. So let's do that. Let's say that the blue chromosomes, let's say those are from dad, and the yellow ones, let's say those were from mom originally. The beads, you'll notice I had different colored beads on those chromosomes. Those represent genes or sections of DNA that code for traits. And then the individual colors of the beads will represent different versions of the trait. And you'll see what I mean when I get down here. Now I had 2 homologous pairs of chromosomes. I had the big pair right here, and I had the short pair here and here. Let's say that the big pair of chromosomes, which had 2 sets of beads, let's say that for, let's say the first trait happens to be, I don't know, let's say eye color. let's say black codes for, I don't know, this could be kind of ugly, but black eyes, and green, maybe that codes for green eyes. And we see that when we look at our homologous chromosomes, these chromosomes here have the black beads, so they would have black eyes. They would give this trait to offspring or children, whereas this one maybe has the green eye trait. They both have the same gene, which would be for eye color, but the two have DNA that would code for different colors of eyes, which is perfectly normal and acceptable. Now, the second trait on those, I had purple and orange beads. Let's say that this trait happens to be for, I don't know, hair color. And purple, let's say that will code for purple hair, orange, orange hair. Not that that would really occur, but for the sake of our argument. And then for the short homologous pair, again, there was two traits. Let's say the first trait is for skin color. And we'll say that blue will be for blue skin, green will be for green skin. And the second trait, maybe, I don't know, being kind of creative here, but fingernail color. And pink fingernails. And the red will code for red fingernails. So now that we kind of have identified the traits and with the beads, let's again take a look. We can see that we have homologous pair. They have duplicated already, which now means that these are in prophase. We can see that we have our red beads matching up, our green ones, so on and so forth. So in prophase, what's going to happen is, just like in prophase for mitosis, the chromosomes, they condense, they're no longer stringy, and they've been duplicated. They kind of take on that X shape. Now in metaphase, it's going to be a little bit different moving forward than we've seen in mitosis. In mitosis, the chromosomes would line up just like this. They would line up in a vertical line. But in meiosis, we are going to line these up kind of together with their homologous pairs. So now we see metaphase. We see that the homologous pairs of chromosomes, they match up and they line up together in the middle of the cell. Now we would have at this point in a real cell 23 pairs of chromosomes. Additionally, they get really snug and we have some crossing over going on, like so. And that's because the cells are very, very small, of course. So we've gone from prophase, now we're in metaphase 1, because this is going to go through twice. And we know what happens in anaphase, same thing as in mitosis. In anaphase, we're going to separate these. But something happens as we do as a result of this crossing over. When we separate these two homologous chromosomes, the sections of DNA beneath the point of where the two homologous chromosomes have crossed over, which would be right here and right here, all of the DNA beneath it, it is going to be traded. So as I start to separate these, I actually have to swap this purple and this orange bead. And as I do so, you should start to recognize that now these two chromatids, they're not identical, and these two chromatids, they're no longer identical either because, while they have the same gene, they have a different version of the gene. Same thing's going to happen with this homologous pair here. We're going to separate them in anaphase, trade some DNA as a result of the crossing over. And now we're in anaphase. We start to see the homologous pair going to separate sides. Telophase, the cells move, or the chromosomes move further apart. We have cytokinesis going on. and we can snip our cell into 2 cells. And what I have just now created are 2 separate cells that are haploid. This no longer has 2 copies of a chromosome. Those are both different. Neither does this one. So now what we've done is we separated this chromosome from this one. In essence, this chromosome here is no longer in the same cell as this chromosome here. And same with this one and this one, and this one and this one. We have divided up all of those chromosomes. Of course, they kind of look like this at the moment. But now that we've gone through meiosis one time, we've gone through meiosis 1, now we need to do the process again. So these 2 and these 2, they would immediately go into prophase 2, followed by metaphase 2, followed by anaphase 2. Now we're separating our sister chromatids. Followed by telophase 2, cytokinesis again, and as a result, I've taken one cell with a full set of DNA and I've created 4 cells with only half of that DNA. Now, what you should definitely take note on and really kind of notice is that while I have 4 cells here, I'm going to get rid of the yellow string just to kind of make it easier to focus, is that no 2 cells have the exact chromosome combinations. They'll all have a chromosome number 1. They'll all have a chromosome number 10. But no 2 chromosomes are the same. For example, this one here, chromosome 1, they both have a green bead. That's the same. But this gene down here, it's different. Same thing between these 2. Different beads up here, or same beads up here, but a different one down here. Those 2 chromosomes, they are different. And the same can be said between these 2 and these 2. So, which one of these 4 haploid cells might be used in fertilization, which would then create an offspring? It's really a flip of the coin, because only one of these will be used. And this is what allows parents to have children that look very similar, because they'll have some common traits, but different because some traits will be different. Now, this is a very simplified process. We're only using 2 homologous pairs of chromosomes, but in the cells of our body, there's 46 of these that this is all happening between. And crossing over, it's completely random. Maybe it happens, maybe it doesn't. Maybe one gene is traded, maybe 2 are. It's completely a flip of the coin. It's completely random, and it never happens the same twice in an entire lifetime.